I got interested in biology after sort of learning about all of the advances in DNA and molecular biology when I was in middle school, and since then, when I was a student in high school, I began to volunteer in a gene therapy lab, and that's where I really began to get excited and get inspired by all of the advances with DNA double helix and cloning and all the recombinant DNA technologies that were being developed and being applied, um, uh, back then. And so this concept of genetic medicine has really been something that has motivated my work and my research over the past 2 decades. So this idea of genetic medicine is really powerful because it provides a modular framework for developing medicine. I put here on the left side a picture of a rocket delivering a satellite into outer space. And this idea of genetic medicine is really, in the same way, a modular approach where you have a vehicle like the rocket, and there can be a number of different payloads that can get delivered by these different vehicles. So they can target different tissues, different cells, and to be able to go into those cells to make the desired modulations. It sort of provides an overview of the two vignettes that I wanted to tell you about today. The first one is on developing human capsid forming proteins and harnessing them for molecular delivery into cells. And then the second story is on new RNA programmable systems that can be used to target DNA or RNA inside the cells. So let's start with the first vignette. Um, over the past 3 years, we have been living through a pandemic, and so viruses have been very much on the front page of newspapers and on TV. And so there are two types of RNA viruses. What we heard about in the news and have been really changed, have dramatically impacted the world, are these RNA viruses like coronavirus or influenza virus. They're RNA based, but they do not insert their DNA into the host genome. So when the host clears the infection, any trace of virus in terms of genetic information is gone. The second type of RNA virus are retroviruses. They include HIV viruses. They are RNA based, but during their replication cycle, the RNA gets reverse transcribed into DNA and then subsequently is integrated into the genome. And so that means the virus infection leaves a copy of the virus's genetic information in the infected host. And so it's the retroviruses that I wanted to talk about today. More than 10 years ago, there was a computational study by Campillos and colleagues, and they looked into the human genome to look for genes that share a significant amount of homology to the structural gag protein of retroviruses. And they found almost 80 different human genes that have conservation with the gag capsid domain. And so what was really exciting is that in 2018, just 5 years ago, a couple of studies published in Cell reported that one of these gag homologs in the human genome called ARC makes virus-like particles. And so that was a really exciting study. And after reading that, I thought it would be really interesting to begin to explore these different proteins in the human genome to see, do they all form capsids? And if they do, can we somehow take advantage of these capsids to be able to deliver molecules to cells? And because they are endogenous to the human genome, they may not have adverse immunogenic effects. And so we studied a number of these different proteins. Early on, we studied a protein called PEG10. And then most recently, the story that I want to tell you about today is the study of a different family of proteins that are also GAG related. They're called PNMAs. They stand for paraneoplastic MA antigen proteins. In the human genome, as you can see on this phylogenetic tree, there are a number of these different proteins, PNMA 1, 2, 3, 4, etc. And what is really clear is, based on sequence analysis, is that they all carry the capsid domain of the retroviral gag protein. And so given that ARC is able to form capsid and PEG10 is also able to form capsids, we thought that maybe we can study this PNMA family and see whether or not they also form capsid proteins. And so what we did is we introduced these different proteins into human cells. We usually use HEK cells as our workhorse. And so by transfecting PNMA 1, 2, 3, 4, etc. individually into HEK cells, we look to see, are there any virus-like particles that are released by the transfected cell? After looking at all of these different PNMAs, we found that there's one specific one called PNMA2, and that is selectively secreted into the extracellular media. And unlike PEG10 and many of the other virus-like particles made by these endogenous human gags, PNMA2 secretes as a non-enveloped virus-like particle. And so what you see here on the bottom two micrographs are EM images of these PNMA2 particles. They are about 22 to 25 nanometers in diameter, and they are non-enveloped, and they look like any virus capsid that you may find. So the next thing we wanted to understand is whether or not PNMA2 package RNA. And so we took the extracellular fraction, we concentrated them, and we looked to see, are there specific RNA that are packaged in the VLP fraction. So whereas you can see the control, which is the cell lysate, shows pretty robust expression of PNMA2 mRNA, the VLP fraction, the things that are secreted by these transfected cells, they do not have any specific RNA that are enriched. And so this is quite different than what we observed with PEG10. PEG10 packages its own mRNA, but in this case, we didn't see any specific RNA packaging by these PNMA2 particles. So given that, we thought maybe we can biochemically study these particles more to see, does it package anything at all? And so one of the things we did is that we expressed the PNMA2 protein in E. coli. So this is heterologous expression. And what we found is that when you purify the heterologously expressed PNMA2 protein, they are not monomeric. In E. coli, they already form capsid-like structures, as you can see here in the micrograph on the left side. We then found that if you reduce the salt in the solution, these capsids will break down into individual monomers. So basically, you can disassemble these virus capsids by dropping the salt in the solution. And so when this happens, you can further purify. So any E. coli contaminant you can remove from the dissociated protein mix. And then after that, you can reassemble. You can just introduce salt back into the solution, and these monomeric proteins will reform the virus-like capsid. So that was really interesting. And so given that we can make large quantities of these sort of protein capsids, we use cryo-EM to solve the structure. And so here is a cross-section of these virus-like particles showing the electrostatics on the exterior and also the interior. And so what you see here is that the interior lumen of this virus-like particle is very negatively charged. So red is negative charge and blue is positive charge. And so after seeing this, everything made a lot of sense because the inside is negatively charged, it's very unlikely that this particle can package negatively charged nucleic acids. And so this natural wild-type PNMA2 probably doesn't package any nucleic acids given the way that it's formed. And so based on that, we thought maybe we can engineer the interior surface charge. And so we looked at the PNMA2 wild-type protein, which is shown in domain diagram on the top. And the part that is negatively charged is in the C terminus and is a disordered region. And we did some protein engineering. We cut off this disordered region, and what we found is that basically that disordered region is not important for capsid formation. So even if you remove it, you can still form these capsids. And so we thought then if we can remove it, maybe we can replace it with something that is positively charged. And so we took the CCMV virus RNA binding domain and we inserted it to the C terminus of PNMA2. And so we call this the engineered ePNMA2. And this modified protein can also form capsids, and having positive charge on the interior lumen now allows us to package mRNA inside of these PNMA2 capsids. And so based on that, we were then able to achieve mRNA delivery using these particles. These are preliminary data, but basically, if you take the engineered PNMA2 and you mix it with Cree messenger RNA, you can treat it with RNAs to make sure that you got rid of everything that is not packaged inside of the capsid. And when you put this onto a reporter cell line that has a Flox GFP reporter, we were able to see sort of expression of GFP. And on the right side is a positive control where we directly, using a liposuction, transfected the Cree mRNA, and you can also see activation of green fluorescence. And so this allowed us to sort of get some early results to show that we can use this in vitro assembled protein particle to be able to deliver mRNA and also be able to functionally express a protein. So we're very excited about this approach, and there's still a lot of work to do to make the system work more robustly, work more efficiently, and also be able to engineer this so that we can target different tissues and different cell types. So anyway, so hopefully next time I can tell you more about our progress with this direction. I'm going to switch gear a little bit to the second story, which is about the exploration of RNA programmable systems. And this is really an extension of a lot of the work that we have done with CRISPR systems. CRISPR systems are very exciting because they are RNA-guided systems that can be developed for DNA targeting and also for genome editing applications. So there are two types of CRISPR systems. They are called Class 1 CRISPR and also Class 2 CRISPR. Class 1, you don't hear as much in scientific literature because they are more complicated, and because of that, they have not been widely used for genome editing. And that is because this system requires multiple protein molecules that have to come and assemble into a multi-subunit effector complex. So it's kind of complicated. Class 2 systems are much more broadly used. So Cas9 is probably the most prominent example of a Class 2 CRISPR system. It's simple. It just has a single protein, and it works with a non-coding guide RNA to be able to target specific sequences in the genome. And so Cas9 has been widely used and is now enabling a lot of different applications. And so some of our early work led us to look at Cas9, but then also beyond. So we found that there are other RNA-guided DNA targeting CRISPR proteins. They're called Cas12, and there are also RNA-guided RNA targeting proteins. They're called Cas13. And so Cas9, Cas12, and also Cas13s, they have all been engineered and developed for both in vivo, so in cell DNA and RNA targeting applications, and also Cas12 and Cas13 have been developed for diagnostics applications, including detection of SARS-CoV-2 and influenza viruses. So after looking at this, one of the questions that came to mind is, where did all these really amazing proteins come from? Where did they evolve from? And to answer that question, two of my lab members, Han Altechuan and also Sumi Kanan, took on a bioinformatic analysis of all of the protein sequences in public databases to see can we find the ancestors of Cas9. So what they did is they took the key signatures of the Cas9 protein, namely the nuclease domains of Cas9, and what they did is they looked in all of the proteins to see are there other proteins that carry a significant overlap of these signatures. And so those proteins turned out to be a family of enzymes called IscB, but the function IscB has not been revealed. But if you look at a phylogenetic relationship here, so this big circle on the left side is the phylogenetic tree. What you can see is that Cas9 is this blue slice here on the bottom right. And the Cas9 protein is one of a very specific branch that emerged from this large diversity of IscB proteins. And so we were then very intrigued by what these IscB proteins might be doing. And so by looking at these IscBs and really looking deep at things that may be the closely related to Cas9, specifically the branches that are most adjacent to the Cas9 branch, we found that some of these are likely to be Cas9-like because they carry CRISPR-like repeats. And so that really led us to look at these systems more carefully, try to figure out is IscB like Cas9 also an RNA-guided DNA endonuclease. So here on the top are the domain comparisons of IscB and Cas9. On the left side, you can see this is IscB. It's got the RUBC1 and 2 and also 3 nuclease subdomains. And then there's also the HNH nuclease domain. So this is IscB protein. And this organization is largely the same as Cas9, except it doesn't have this recognition domain that is inserted between the bridge helix B and also the second RUBC nuclease domain. So Cas9 is known to work with non-coding RNAs, CRISPR-RNA and tracer RNA. And so based on the similarity, we asked whether or not IscB could also have a non-coding RNA working with it. So we did some RNA sequencing. We took one of the bacterial strains that carries IscB, this K. racemifer bacteria, and we did RNA sequencing, and we looked at non-coding RNA that are expressed in the vicinity of IscB. What we found is that there is a very robustly expressed non-coding RNA region right upstream of IscB, and part of this is very highly conserved among different IscBs. But as a part of the transcribed RNA, there is also a non-conserved region. And this is what we hypothesize to be the spacer, the guide sequence that directs IscB to specific positions in the genome. And that turned out to be exactly the case. And so we tested IscB, and we showed that IscB is exactly functioning like a RNA-guided Cas9. And so we then solved the cryo-EM structure for IscB. And you can see based on the structure of IscB and comparing that with Cas9, IscB is very small in the protein component. So these yellow parts are the protein. Cas9 is much larger, and the rect domain that is missing IscB is the orange yellow domain that's on top over here. What is very interesting is that this rect domain protein part in Cas9 is replaced by non-coding RNA domains in IscB. And so what really happened is that IscB, the ancestor of Cas9, was a small protein working with a very large non-coding RNA to form this ribonucleoprotein complex. As evolution took its course, a lot of the non-coding RNA got replaced by protein domains to make it into a more robust RNA-guided nuclease that bacteria can use as a part of their immune system. So this sort of led us to understand the evolutionary origin of Cas9 a lot better. And the compactness of IscB potentially has useful applications for in vivo delivery because the viral vectors or the nanoparticles that are used to deliver these things usually work better when things are smaller. So this is one potential direction to further develop IscB. In addition to IscB, we also found the ancestral protein for Cas12, and that's a protein named the TMPB. TMPB is really interesting. It's probably the most abundant protein in all living organisms. In a single bacterial genome, you can find sometimes 500 copies of TMPB. So they are as abundant, if not more, than ribosomal genes. And CRISPR-Cas12 really came from this really broadly occurring protein. Another interesting thing that we reported recently was that there are CRISPR-like systems, TMPB derived, that are found in eukaryotes. Sometimes you can find them in single cellular eukaryotes, and then you can also find them in some cases, more rare, multicellular even animals. So this clam here has these RNA-guided systems inside. And these systems are called Fenzor. And if you look at a cryo-EM structure for Fenzor, they look very much like the Cas12 proteins, and also very much like the TMPB. They have a protein domain with a large rect domain, as well as a large non-coding RNA that we call omega RNA to be able to direct this to recognize and also cleave specific DNA sequences. So Cas9, Cas12, Cas13, IscB, TMPB, Fenzor, PNMA2, etc. They're all naturally occurring systems that provide very important functions in their native context. And what this really highlights is that if we further explore nature's diversity, there will likely be many more interesting systems that can be co-opted and engineered to do something that's useful to improve human health or improve other biotechnological applications. And so we're very excited to continue to explore that. This plot here shows the really rapid exponential growth in the amount of biodiversity genomic information that's being collected. And I think it's really the exploration of this that we're going to find a lot of the powerful tools that nature has developed that are encoded in these DNA double helices. So that's all for today. I just want to thank you again for the opportunity to share our work with you today. And also I want to acknowledge my team at MIT, as well as collaborators and funding support that has enabled us to do this research. Thank you very much, and I'm happy to answer any questions.
Thank you.